Hey guys, so today we're going to continue our discussion of evolution. We're going to focus on speciation and phylogeny. I know you guys love your phylogenetic trees. All right, so here we go. I want to remind you guys about the modern synthesis theory, or the idea how we are going to link Darwinian evolution to an understanding of genetics and modern understanding of inheritance. So we use what we now know, our technologies that are far more advanced, to sort of put that together with our ideas that Darwin had. So we can now define evolution not only as change over time, but changes in allele frequencies over time. So variations in traits that are a result of the changes to genes known as mutations. So alleles are different variants in traits, and we can now define evolution as this. So keep that in mind. Also, do not forget, I said this in my last video, evolution is a population level phenomenon. Individuals do not evolve. The evolution of a population emerges from the individual fitness of members of that population. So whether or not individual members will survive and reproduce and pass those traits on to offspring, that can affect the frequencies of alleles in a population. So as they survive and reproduce or not, the frequencies of alleles in the next generation will change accordingly. So an individual cannot influence the frequencies of alleles in a population beyond the fact whether or not it's going to survive or reproduce. But the changes in frequencies, that's really what evolution is all about, at the population level, not at the individual level. Okay, phylogenetic trees. A tool used by scientists for comparing evolutionary history or relatedness among species is called a phylogenetic tree or a cladogram. Now, I have this whole other video on cladogram. I think it's one of my more popular YouTube videos, and it goes through and kind of explains step by step how to read and create a cladogram, so please check out the cladograms video. I'm going to briefly go through how to create a cladogram or phylogenetic tree on your own here, and then we'll do some practice ones. So the number of characteristics that are shared among the different items you're working with are going to be determined first. And to do this using a character table works well, and so I'll show you what one of those looks like later. And then you arrange the items in a tree diagram to represent these similarities. So I have my whiteboard back here, but you will start with a common ancestor up here and then go all the way up. That marker is terrible. To your terminal taxa or group here, and you can have branching here and traits here, and we'll talk about what those are in a little bit later. But the idea is that we want maximum parsimony. So arranging the organisms in a tree diagram showing the simplest possible evolution. You don't want to have a bunch of traits evolving at different times. Ideally, you would mark that trait down here only evolving once, because the simplest solution is often the most correct one. Okay, so according to the cladogram, which features do lizards have? And that's easy because we look at where the trait for lizards, we look at where the organism lizards appears, it's here, and then we look at the traits that precede it. Now every organism that comes after where the trait is marked, so all of these organisms would have claws or nails. So lizards, pigeons, mice, and chimps are going to have claws or nails. So the lizards will have everything that precedes it. So lizards will have claws or nails, lizards will have lungs, and lizards will have jaws. So it would be D on this particular one. Oops. Okay. Here's another one. Which two groups of organisms have the most genetic differences? Okay, pause it, try to figure it out on your own. I'm going to reveal it. Okay, it's mollusks and arthropods. Now, compared to the other options, those ones are the two that are furthest apart. They have the most branching in between them, so they're probably going to have the most genetic differences. They'll probably be less related than the others that are closer. Okay, a tree diagram is meant to represent an actual history of evolutionary lineages, but not necessarily to scale time-wise. This could occur over millions of years, but how they are related and branch over time. Now, these four trees all depict the same evolutionary history, so you want to be careful. Sometimes the order doesn't matter as much as the branching pattern, so always pay attention to your branching pattern when you're creating phylogenetic trees. Some important terms that we want to use to describe our phylogenetic trees are the tips, the ends. Generally, these are going to be representing species, but sometimes groups of species. So the taxa are called the tips. These are the ends. An external branch, this is at the end. A node is where we would have a common ancestor, or where we have a different branch off, and then we have internal branches here, and of course our root is going to represent our common ancestor to all of these organisms that follow. Okay, we also see that traits appear. We're looking at how these trees show evolutionary history. If a trait appears here, all the organisms that follow will have that trait, and nothing before it that precedes it will have that trait. Okay, our phylogenetic trees can get very complicated. They can show many different traits and many different types of organisms. It just depends on how much information you have and how much you want to include. Okay. Most of the species concepts proposed by biologists are different ways of approaching the question, what are species? So, oops, there we go. We know that a simple definition for species is a group of organisms that mate with one another. And speciation is the divergence of biological lineages and emergence of reproductive isolation between lineages. So let's talk about reproductive isolation. Well, first of all, let's talk about these different species concepts. So a morphological species concept is just the idea that organisms look alike are going to be members of the same species. And Linnaeus came up with this. He also came up with our binomial nomenclature, which is the idea that we classify species and then they have a scientific name that includes two words, binomial. So for example, Homo sapiens for us, or Gallus gallus for the chicken. And there's tons of them out there, and we continue to use very similar rules to the ones that Linnaeus came up with. Now, the biological species concept is a different concept of what a species is, and that's the idea that species are groups of actually or potentially interbreeding natural populations that are reproductively isolated from other such groups. So they only could breed with that other members of the species if they're brought together. Now, reproductive isolation, I don't want you to think that this means that they're physically separated, because you can have reproductive isolation of a species when they're in the same place. But it just means that two groups of organisms can no longer exchange genes. This is a good example of organisms that may be morphologically similar, but they're actually not part of the same species. So that's why we cannot always go with the morphological species concept. These are two different frogs. You notice even their coloring on their backs looks very, very similar. However, these are actually species that do not interbreed. They do not reproduce and create viable offspring. Okay, speciation can be punctuated, it can be in bursts, or it can be very, very slow. So the development of reproductive isolation may take millions of years or may develop in a few generations. So there's some bats and Drosophila in the lab. We often see this, but sometimes this actual branching pattern is going to take a very long time. Okay, one type of speciation I want to talk about is called allopatric speciation. Now this is when populations are separated by a physical or geographic barrier. They actually are separated. And then because these barriers can form when continents drift or when sea levels rise and fall, when glaciers advance and retreat, and climates change. And then what happens is when these two are physically separated, we have changes in alleles and they're no longer physically mating. And then if they are ever brought back together, they are reproductively isolated and they can no longer exchange genes with one another. They can no longer reproduce. Now, sympatric speciation is going to be speciation without physical isolation. And this can occur with disruptive selection sometimes, where individuals with certain genotypes prefer distinct microhabitats where mating takes place, so that could be the cause of separation. But sympatric speciation most commonly occurs by polyploidy, where we have a duplication of a set of chromosomes within individuals. And this is very frequent in flowers because flowers or angiosperms self-fertilize. So speciation by polyploidy is common in plants, so about 70% of flowering plant species and 95% of fern species, actually. So there's lots of different types of speciation. Again, we're focusing on allopatric and sympatric. Peripatric and parapatric are less common. Parapatric is actually very rare, but it does exist. But I do want you to focus on the first and the last one. So right here, the one thing to remember, the big difference between allopatric and sympatric. Allopatric has that physical separation of, or a barrier formation between the two groups of the same species. But in sympatric, we're going to have some sort of genetic change, generally polyploidy, and then we end up having some sort of reproductive isolation occur, and we have two new distinct species that cannot exchange genes. Okay, we're almost done. I want to go through a few practice questions before we finish up. So these are based on a bunch of different things, some of the evidence for evolution, some of the general evolution stuff. They're pretty easy though. The figure below illustrates how different plants can have similar structures that are modified for different functions. What is this an example of? Take a second to read the answer choices. Homologous structures. Now, remember, we see this example a lot with the bones in like a bat and a human, but homologous structures are similar structures modified for different functions. Okay, which of the following best describes an adaptation? Go ahead and pause it. You can read them. Okay, is a trait that occurs within a population that is advantageous and thus increases fitness. Perfect example of an adaptation that was just a memorization question. Okay, Which is an example of evidence for evolution based on comparative embryology? This one is so easy, but pause it. Okay, human embryos have gill slits in early stages of their development. Okay, so obviously this one is super easy because it has to do with embryos, and we're asking which one is based on comparative embryology. All of these, though, are different types of evidence for evolution. Okay, upon close examination of the skeleton of an adult python, a pelvic bone and tiny leg bones can be observed. These features are examples of, you got it, vestigial structures. Now remember, a vestigial structure is potentially a structure that is retained after many years and many divergence and evolution, but no longer has a function. So some people say that the appendix in humans is a vestigial structure. Maybe it used to have some sort of ancestral purpose, but in modern humans it no longer does. Now there's some scientists that disagree with that. They say yes, there is actually a function for the appendix, and we think it's this, and this. However, most people agree that there are vestigial structures within humans, and that could be one of them. All right, so that's it for this video. Please make sure you go back if I went too fast, and I'll see you later.